Hello everybody, welcome back to microbiology. Today we'll be going over the cloning lab. First, let's talk about some basic characteristics of plasmids, also called vectors in molecular biology. Plasmids are circular DNA. They have extra chromosomal, or in other words, accessory genes such as antibiotic resistance, or genes that help them handle certain environmental stresses but aren't necessary for their basic cellular functions. They can be exchanged during conjugation, or they can also be inserted, in our case transformed, into cells in molecular cloning. There are some essential features that are necessary for a plasmid to be used as a cloning vector. The first is an origin of replication. We've discussed the origin of replication in DNA replication in cells. It's also important for plasmids to contain an origin of replication. This is a specific DNA sequence that allows plasmid replication to begin and take place. The next is a selectable marker. They usually provide resistance to an antibiotic. And in doing so, we can select for only cells that have those antibiotic resistance genes. In this case, we have the antibiotic resistance gene for ampicillin, labeled as AMPR. Because AMPR is present in this PUC19 plasmid, if we were to transform with this particular cloning vector, only E. coli cells that contain PUC19 would become resistant to ampicillin. So after transformation, the bacteria can be plated onto a medium that contains the antibiotic ampicillin, and we will only have E. coli cells grow if they have that plasmid present. So in this way, we can select for just our transformants. Lastly, we have a cloning site. The cloning site is the region where we can insert the genes that we're interested in. So this is kind of the business end of the plasmid or the cloning vector. It has all of the necessary components to make this happen. It has a promoter here. This is the lactose promoter. It has an operator, the lactose operator, and a region that contains restriction enzyme sites we'll talk about soon here. So in the last lab, we worked a little bit with restriction enzyme sites. We had to design primers that included restriction enzyme sites. And we said that we would discuss in greater detail exactly what they do. So restriction enzymes actually catalyze, or they allow the breakage of the phosphodiester bonds in the DNA backbone. Each restriction enzyme has a specific DNA sequence that acts as an address for that restriction enzyme. If the restriction enzyme finds that particular DNA sequence, it will cut both top and bottom strands, the same base pairs, in order to allow separation. So let's see how this looks. Here I have presented you with one of those restriction enzyme sites, or the address for the restriction enzyme. This particular enzyme, EcoR1, has a restriction enzyme site GAA-TTC. Here we have the top strand, 5' to 3', and the bottom strand, written in the reverse direction, 5' to 3'. You'll notice it's the same sequence, GAA-TTC. And so what happens, as I said, the restriction enzyme is going to cut at that same base pair on the top and the bottom strand. So it's going to cut between the first G and A and the first G and A, reading 5' to 3'. You don't need to memorize where these sites are exactly, but you do need to understand the mechanisms of them. So if we take that site again and we snip those two regions, you can imagine then that we're going to get a separation of that region there. So if this was a linear strand of DNA, it would become two fragments. If this was a circle of DNA, or a circularized plasmid, it would become a linear strand. Imagine if you had a rubber band and you cut it in one spot, it would just become a straight band of rubber. And if we go ahead and fill it in, we can see that if you recall that the 3' end always has a hydroxyl group, and the 5' end always has the phosphate group. So we have our phosphodiester bond that gets broken between the 5' end and the 3' end. And while we're here, we might as well see that ligase does the reverse of this reaction. So it's actually going to take those two strands and can ligate that phosphodiester bond back together, or seal the nick of that phosphodiester bond breakage. This is a typical experimental setup for a restriction digest. The ingredients for all of these come in their own tubes, and each one of these ingredients is added. So for a restriction enzyme, you would add what they call units. We won't go into detail about what that means, but it's a certain measurement of how much restriction enzyme is in a certain volume. And you would add 0.5 to 1 microliters. For your DNA, you want to have somewhere between 0.1 to 4 micrograms of DNA. Buffer, about 5 microliters. And then water, you're going to top off, so your total reaction volume is then 10 to 50 microliters. You don't have to memorize any of these units. I just wanted to show you how this is usually set up in a test tube. Okay, the next enzyme we're interested in is the ligase enzyme. And as we've talked about before, it will repair the broken phosphodiester bonds. So we've cut with the restriction enzymes, and this process allows us to paste two DNA sequences together. So you can see right here that we have the phosphate group on the 5' end of the ribose sugar, and on the 3' end we have the hydroxyl group. And this phosphodiester linkage is created between these two nucleotides. So now let's put this all together. In the previous lab, we needed to amplify a gene of interest via PCR, and we designed primers that included restriction enzyme sites on the ends of each side of that gene. So in step 1, we can see that a gene of interest, in our example this was GFP, was amplified by PCR, and the plasmid contains the restriction enzymes on either side of that gene of interest. Step number 2 is we do a digestion of the PCR product, or our GFP, and the vector or the plasmid where we want to insert it. So we'll be cutting the plasmid at that multiple cloning site. So then we can ligate in step 3 our amplified and digested gene. Here, these little blue Pac-Man symbols represent the restriction enzymes. So our amplified and digested gene, we can then paste into the digested plasmid if we use the same enzymes on both the plasmid and the gene of interest. And then lastly, we'll have our transformation, which we haven't really talked about yet. Okay. So, and you can ignore this dephosphorylation step. It's not important for the type of cloning that we're doing. So let's talk about bacterial transformation. This is different than conjugation, where conjugation is when two bacteria share DNA by coming into direct contact with each other. Transformation is the process of taking up what we call naked DNA, or DNA that's not in a cell. And in order to do that in the lab, we have some specific steps we need to follow. Sometimes this can occur in nature with naturally transformable bacteria. So you have a recipient cell and you have your naked DNA. The naked DNA is taken up by the recipient cell. You have genetic recombination, where the naked DNA is assimilated into the chromosome. And then we call that a transformed cell because the phenotype or genotype has been transformed by this process. Now, we typically use E. coli to do these transformations because E. coli is a very easily culturable bacterium. It's the most well studied of any bacteria out there, and it's really good at producing genes and making compounds in industry that are useful for us and economically viable. And so it's a really good host for this process right here. So what happens in the lab is bacteria do not naturally take up DNA. So scientists make the E. coli competent by altering their cell wall and membranes. The reason why this is necessary and the reason why this works is because the cell wall has an overall negative charge, and this repels the negatively charged DNA backbone. The cell wall has phospholipids, and those phosphates are negatively charged, as well as LPS, and LPS is also negatively charged. And if we look at the DNA backbone, we see this ribose group and this phosphate group. And if we look carefully, this phosphate also has a negative charge. So these two negative charges naturally repel each other. So we need a way of neutralizing these two charges so that they can come into close contact with one another. So this is what we mean by making E. coli chemically competent. In order to make E. coli competent, we can do so by the addition of the chemical calcium chloride. Calcium chloride neutralizes the repulsive negative charges of the cell wall and membrane of the phosphates in DNA. As you can see, calcium from calcium chloride is positively charged. So when it interacts with a phosphate group, it neutralizes this negative charge. And this allows the DNA and the cell to come into close contact with each other. Otherwise, they repel each other like two negative poles on a magnet. So after the cells are made chemically competent by the addition of calcium chloride, the DNA is added to the cells, and then they are what we call heat shocked. Heat shocking is done by incubating at 42 degrees Celsius. Remember, E. coli likes to grow at body temperature, which is 37 degrees Celsius. By increasing the temperature above their optimal growth rate at 42 degrees Celsius, this increases the cell permeability to DNA. So if you imagine if you heat up butter in the microwave, it becomes liquid. So it becomes a lot more easier to pass molecules through it because the membrane becomes a lot more fluid. And also because of the heating, you can create pores in the bacterium cell wall where convection currents run through. It's not necessary to memorize and digest all of that information, but you should be aware that heat shocking at 42 degrees Celsius does increase cell permeability to DNA. Lastly, the cells have had a rough time from this. They're made chemically competent by adding a bunch of calcium chloride. We heat them to this 42 degrees Celsius, and so now we need to go through a step called recovery. In this step, naturally, cells sort of recover from this harsh treatment, but maybe more importantly, it allows them to begin to express the proteins or the genes that lead to proteins that are present in those plasmids. And the most important one to begin expressing right now are those antibiotic resistance genes. Because remember, we're going to go from the transformation to putting these on plates that have those antibiotics present. So we need to make sure that they become resistant to those antibiotics before we plate them onto them, or else even though they have the plasmids present, we'll end up killing them. All right, so now we have our selection step. So after transformation and we've done our recovery step, the cells are plated onto a selective medium. The medium often contains an antibiotic to which the plasmid confers resistance. And the E. coli cells are incubated at 37 degrees Celsius overnight, which gives them long enough for little colonies to start growing up. As we can see, here are the colonies. If there's no antibiotic present, this would grow in a giant lawn. But because the antibiotic is present, we only see colonies appear from a single cell that had the antibiotic resistance genes present. Okay, we've come a very long way. We PCR'd our gene of interest, including restriction enzyme sites on the ends of that gene, the flanking regions, that match up with restriction enzyme sites in our plasmid, so that we can take our gene of interest and our plasmid and do a restriction digestion with our two enzymes, and then we can ligate them together. Then we followed that up with a transformation and a selection. So now that we only have bacterium that have our plasmid present, now we're ready for expression. And what determines our expression was already done earlier on our plasmid vector. As we said, for our example, we had the lactose promoter and lactose operon present. But there are a few different ways to do this. We could design it three different ways. We could design that gene of interest to be constitutively expressed, and that means that the expression is always on. So the genes are always being made. It doesn't matter what's going on in that cell's life. The genes are always going to be expressed at about the same level. We have a repressed expression or a repressible expression, where the expression is usually on, but it can be turned off under certain environmental conditions. And the gene expression could be induced or an inducible expression, which is a situation that we're going to be looking at where the expression is usually off, but it can be turned on under certain environmental conditions. This is a very common way for doing expression in expression vectors because you don't always want your genes to constantly be expressed. Sometimes these genes are toxic. Sometimes you're expressing a lot of this gene and the cells just can't handle it very well, so it makes them unhealthy or sick. And if you're going to be expressing something, sometimes it's good to do it just when you want to do it so that you can measure the effects of it once you turn it on. And it's also great that if you're trying to harvest a product, that you can express all at once and gather a whole bunch of your product at once. And that way you're not trying to slowly, milk the cells of that particular protein or product. So an induced expression system is probably the most common expression system you see on vectors today. This is the expression system that we're using in our plasmid. It's called the Lac Operon. We've already touched on this in lecture, but let's revisit it here. So this is an inducible expression. So the system is usually off, but can be turned on. We have a promoter here. It's just labeled LAC-P behind the RNA polymerase. Remember, the RNA polymerase will transcribe a gene into RNA, into messenger RNA specifically, and that's destined to go to the ribosome and be read and translated into a protein. This RNA polymerase, however, is stuck behind this control region. Here's this Lac operator. This Lac operator usually is like a red light. It represses the ability for RNA polymerase to express itself. It does so through the use of Lac-I, LAC-I expresses the repressor itself, and the repressor identifies the LAC operator sequence and binds to it. And then these are just the genes that could be expressed under the LAC system. We actually aren't going to use any of these genes. You can replace these genes with whatever you want. All we're interested in is taking advantage of this expression system, the LAC-I, the LAC-P, and the LAC-O. We're going to replace LAC-Z, LAC-Y, and LAC-A with our own genes instead. So this can allow for controlled expression of our gene of interest. We can do so by introducing the chemical compound that causes this repressor to change into a green light. So that compound that removes this repressor or turns it into a green light so that RNA polymerase can move along and transcribe this gene is something called IPTG. Allolactose does the same thing, and IPTG mimics allolactose. Allolactose is a metabolite in the process of metabolizing lactose. Obviously, if lactose is present, you want to transcribe the genes that are involved in digestion or catabolism of lactose. So that's why lactose activates this transcription of this pathway. So if you have your cells, you know, we already have them on our selection plates, we put them in a liquid culture, they're not ever producing any of the downstream genes. But when we're ready, when the cells are good and ready, we add some IPTG to the culture liquid, and that gets into the cell, and it causes this LAC repressor to come off of the LAC operator, and then our RNA polymerase can go on through and start transcribing our gene of interest. Let's talk a little bit about our gene of interest. Our gene of interest in this lab, or GOI, is the gene that produces the enzyme pyruvate dehydrogenase. If you recall in our cellular respiration lab, pyruvate can be converted to acetyl-CoA. And this is the first step prior to entering the TCA cycle. This conversion of pyruvate to acetyl-CoA is done by the enzyme complex pyruvate dehydrogenase. What we hope to accomplish in this lab is to make more acetyl-CoA. So, you know, in the cell, carbon can be used for all sorts of different processes. This pyruvate could be used to do a lot of other side reactions and side metabolism. We want to try and drive the pyruvate into acetyl-CoA, and we want to try and increase the rate at which acetyl-CoA is produced, because our particular strain of E. coli also has this other pathway engineered into it. We're calling it, just for short here, the artemisinin pathway. And we want to make lots of this compound because it's an important antimalarial drug. So our goal is to overexpress or express even more of this pyruvate dehydrogenase by putting it on a plasmid and introducing it into our artemisinin producing E. coli strain. Well, to understand how this affects our artemisinin pathway, we want to add another gene here so that we can co-analyze the pyruvate dehydrogenase, because, you know, this isn't to the naked eye or to even an eye underneath the microscope, this is an invisible compound. So we can use something else to help us visualize what's going on. And that is the gene for green fluorescent protein. This is a small protein that was discovered from the jellyfish Aquaria victoria, I hope I'm saying that right. And what's interesting about this protein is when you expose it to UV light, it fluoresces a bright green color. And so it's commonly used in fluorescent microscopy. This gene can be introduced into a cell. Here we have a human astrocyte, which is a cell that exists in human brains. And when it expresses this GFP, it lights up the structures of this cell. And as you can see all of the interesting patterns and networks that exist in the cell that you might not have been able to see under a typical light microscopy image. In this example, the GFP is kind of expressed all over the cell. I don't know what exactly the expression vector is like, but you can do a variety of different things with GFP. For one, you can use it as a reporter gene. A reporter gene is a gene that's commonly transformed with other genes, and its expression can be easily identified, measured, or quantified. So unlike that pyruvate dehydrogenase, where we can't see it, it's not very easily identifiable within the cell. If we add our GFP to that same vector, we can definitely measure how much GFP is being produced by how brilliant green it is. And in that way, we can determine how, by proxy or relatively, how much pyruvate dehydrogenase is being made. So reporter genes can be useful to determine if a gene of interest has been, first of all, transformed. If you don't get any green fluorescence, then you probably aren't really looking at a true transformant. Maybe it's maybe it got antibiotic resistance through some other means, or maybe it's a contaminated sample that has some other bacteria that are resistant to your antibiotic gene. You can also determine the level of expression. The greener the cell is, the more expression you're looking at. And if expression is there at all, right? If there's no green color, then there's no expression at all. And also you can determine where it is expressed. So these genes might only be turned on in certain parts of the cell, like maybe the cellular membrane, for example, or maybe in only certain cells of a multicellular organism. So GFP will only be lit up in the cells that express that certain gene. So in that way it can report back to you, okay, the gene I'm interested in is only expressed in maybe lipocytes, or it's only expressed in astrocytes, or wherever else you're seeing the green color in certain cell types. So our cloning lab, after you finish watching this video, you're going to read the introduction, and following the introduction in the document, you'll find a protocol for simulating cloning of the GFP gene into your plasmid. You will need to use your GFP sequence that you previously made in the previous lab. So make sure you have that handy. If you're using a different computer, go ahead and send that to yourself and download it onto the computer you're currently using. After that's done, you can take the online Canvas quiz and you're all done. So this is your cloning lab instructions. Here it just tells you that it's a two-part lab. So you're going to use that map of the GFP you made in SnapGene, and you're going to continue using SnapGene for this lab to simulate your cloning techniques. And then when you're all done, you can take the quiz. Here's a section of your introduction. Essentially, it explains how you're going to be using a production strain of artemisinin, which is an important antiviral drug. And so your goal is to increase the levels of artemisinin that's being produced, or the titers of that artemisinin. And to do so, you're going to try and overexpress that pyruvate dehydrogenase enzyme. Again, that's because this compound uses acetyl-CoA as a pre-metabolite in the metabolic pathway for producing it. And pyruvate dehydrogenase could potentially increase your acetyl-CoA pools. This is what your plasmid map is going to look like. It's called lowercase p for plasmid, PDH for pyruvate dehydrogenase, and OE for overexpression. You're basically going to do steps basically 1 through 3 in this lab. So you're going to take your PCR product. Essentially, you can just use your GFP sequence for that. And we're going to assume that it's been PCR'd. And you're going to simulate digestion of that GFP, the digestion of this vector, and the ligation of the two. So if you look carefully here, here's your pyruvate dehydrogenase. It's under the Lac operator and Lac promoter system. That is kind of hard to see, but it's the blue and the white above it here. And you have two restriction enzymes, this AVR2 and XBA1. And that's going to be your cloning region. And you're going to insert, using this traditional cloning paradigm, you're going to insert your GFP just before pyruvate dehydrogenase, and they're going to share the same promoter system. So if GFP is being expressed, pyruvate dehydrogenase is being expressed, and vice versa. And in that way, GFP will act as an excellent reporter gene for the expression of your pyruvate dehydrogenase. As always, feel free to contact me if you have any questions about this lab. And that's about it. Good luck to you all. Bye-bye.